Okay. Thank you for inviting me to give a presentation today, and thank you for the very kind introduction. So today I'll talk a little bit about some of the work we're doing with the way we use polymers for controlled delivery systems in really trying to address some of the problems in global health. So I'll focus on three areas where we've used polymers to really tackle some big problems in the area of vaccines and malnutrition that can really affect large number of populations globally. So I'll first talk about some of our vaccine applications, and I'll switch over to our micronutrient. All of our work is heavily focused on using polymers in these different systems, both as tools to build things, and then also as tools to stabilize sensitive molecules like vaccines and liable micronutrients. So I think I don't need to, I still like to show this slide because the world has changed so much over the last few years. But I used to present this data to motivate some of the work in the vaccine space. We are, one of our very supportive sponsors is the Gates Foundation, and they very much care about the under-immunized populations in low-income countries in the developing world. And this graph, or this picture here on the left, sort of shows an old view of where all of the vaccine preventable deaths were occurring globally. So you can really see that it was in particular countries and wasn't really affected affecting some of the more developed countries. And of course, if we looked at in the United States, the leading causes of death before the pandemic, obviously influenza and infectious diseases were quite low on the list. And of course, heart disease and cancer were the dominant ones. Of course, all of us know this has changed drastically in the last 2 years, and really COVID-19 has put infectious diseases really up there on top of the list and really emphasized how important it is that we develop new tools for vaccinations and really start looking at vaccines as well in how we can improve them in order to help with some of the challenges that exist now everywhere in the world, not just in some of these developing world countries. So with that, this is data from a few weeks ago, just kind of to remind everyone in the case of COVID, at least, because we have such great data these days in this area. Obviously, many cases of COVID, and unfortunately over 6.5 million deaths globally. And this is an interesting, this is all data from WHO. They have this very interesting dashboard for those of you that maybe don't follow this daily, but this is a vaccination plot, and obviously we've given almost 13 billion vaccines, which is just phenomenal when you think about it. And these are a lot of these are novel mRNA vaccines. But what you see here, what really jumps out is that still this area of developing world is hitting the hardest. So the vaccination rates are really low in this area. And so this is kind of the focus of my work, is how we can use our expertise in polymers and controlled release of biologics and stability to really help these areas. So I think some of it is now very much related to what we do today. So I'll start with our work on self-boosting vaccines. So single injection delivery systems. And really the idea here is, can we put, for many vaccines, you require more than one shot, so a prime and then one or multiple boost dose. In the case of polio, for example, you require 3 shots. And can we put all of that in one shot? Can we somehow develop a formulation that allows you to do that from one shot? Because what people have shown is that over time, even in these areas of limited medical infrastructure or healthcare infrastructure, there is some access to a child after birth. And so if one could combine the shots in less, or the vaccines in a single shot or less shots, there might be a higher chance for the baby or child to be fully protected. So that was the idea. And as we think more about today and how this could be relevant to some of the pandemic readiness and issues that we see today, is if you look at the data for COVID, for example, we have very high compliance with the first shot, and then as you go down to second shot and then subsequent booster, you have lower and lower compliance. So the idea is, if we did have something that was a self-boosting shot that had maybe multiple doses that come out from that same one injection over time, that you might reach herd immunity quicker because you obviously have much better compliance. You might also be able to get higher, more sustained titers against the pathogens because you are self-boosted at the right intervals. And then those things would ideally decrease the chances of variants occurring because you had limited residence time of the residents, even if you got infected, because you were protected with the antibodies. So that was the idea. And the really fundamental hypothesis is how we could leverage polymer drug delivery to create something that would allow us to deliver these type of kinetics in vivo. And for those of you that are familiar with drug delivery, most of the drug delivery of biologics is based on a matrix type system, where a polymer, in this case in gray, a biodegradable polymer, PLGA, is what we use here. But if you think about it, the drug or biologic is distributed, which is this pink color, throughout the particle. And so this has been shown by us and by many others over the last 34 years, that if you make these type of formulations when they're hydrated, that is when they're injected in the body, delivered to the body, you'll have this instant release, which is usually shown as a pulse that releases from the surface of the particle, your biologic, and then over time, the pores left behind from that initial burst allow for the biologic throughout that particle to diffuse slowly. And so with that, you can tweak around the formulation and you could get continuous release kinetics. And you can get something like a pulsatile release kinetic that's maybe a little closer to what you would get with subsequent immunization shots. But in this case, you usually have a big pulse, then a quiescent period, and then you have this slow release later, which is governed by this diffusion and degradation of the particle. And while these are very interesting systems and they've been used clinically for different applications, what really is required for an effective immunization, and again, I'm a material scientist, not a biologist, so I won't go into the, or immunologist, into the biological response, but our understanding is from those experts in those fields that really to get your body to develop a strong, robust protection against the pathogen, the immunization needs to occur in pulses, and there needs to be a quiet period between the different pulses. And that really mimics the sort of bolus presentation that you get when you get subsequent shots when you visit a doctor. So our hypothesis was that if we change the structure of the particle at this very, very fine scale, that we could have kind of a core shell structure. If we had a core shell structure like an egg, then the shell or the polymer of this tiny little particle, when that fails, the drug or biologic or vaccine that's inside would then release instantaneously. And it turned out that this was in fact the case. It was very hard to build these particles, and I'll go a little bit into how we did that. But once you make them, you are able to get these very beautiful pulsatile release kinetics. And this work was published in Science in 2017. And I will kind of show you. So we've focused primarily the methods we developed to make and control the structure of these particles at that fine scale, so they're still injectable. We focused on PLGA polymers, which are polylactic glycolic acid. These are polyesters that are biodegradable by hydrolysis, widely used in the clinic as resorbable sutures. So they have and other things, but they have a very good record of safety in infants and children and adults. Very widely used. Most of us have, if we had any minor dental or other type of surgery, I'm sure we have had these sutures used on us. So we use these materials, and we developed methods that were borrowed from the microelectronics industry in really being able to, we ended up making cube structures rather than spherical. So we made these bases, what I call bases. So they're little cube cups that are open, which can be filled with essentially any drug. And that's right now we use a aqueous fill. So as long as you can suspend it, have a stable suspension in aqueous solution, or if it's soluble, you can fill the particles. And then the lid is attached and sealed onto the particle, which forms a full seal. You can see an SEM here on the left, and here's an optical image where you see the PLGA polymer is sort of clear. And inside, you can see that in this case, we filled it with a dye. You can see that you can fill the core. And as if you look here on the right, this is an array. We can make about 1,500 per histology, a little glass slide. So I'll show a couple of videos just to illustrate how this is done. So this is obviously a cartoon-based video, but and it only shows 4 particles, but we do these in large arrays of hundreds to 1,000. So you can see how the bases are molded from PLGA, and then we have a piezoelectric nozzle, which can fill picoliter volumes. Again, these are quite small, in the order of a couple of 100 microns. And then the lids are aligned and sintered to the top. And once that is done, the particles are fully formed with the core holding the drug. The drug is formulated in there in dry format. Another quick video just to illustrate. This is actual optical video of again, a dye is being filled so you could see it, but these are sort of the bases made out of PLGA, and then they're being filled with a blue dye for illustrations. And as you can see how this method could be scalable and automated. And obviously you could fill different things into different particles. And here, again, I want to bring your attention to the bottom particle. You can see there's a diffraction pattern over the particle. This is where the lid is sealed to the particle, which is obviously very important for us. So there's no leakage for a long period of time once it's injected in the body. And you can see we follow it until this diffraction pattern is completely eliminated, and that ensures that it's sealed. And the reason this is important is, one, it gives us an indication of the integrity of the seal, but two, as you think about scaling this, this is one way that it can be automated. You could use optical and AI type sort of a software-based system to ensure the quality of these particles as they're being sealed. And so here again are some SEM images and optical images of the particle. This was our first generation particle. Again, you can see here on the left, it's a particle with a small well. Here it's filled. You can see the dry, once it's dry, the material inside the particle, the lid is sintered on, and you can see a cross section, and you can really see that the lid is integrated with the base. And then here in the second row, especially the middle eye, you can see these are large arrays of the particles. Here they're filled with, again, a dye, and again on the right, they're filled with a red dye, and you can see that the core and the integrity of the core and the drug inside the core is maintained. And so this is example of the ability to manipulate. This method really is based on a mold, so you can manipulate across the top is increasing loading while keeping the size the same. The bottom is keeping the size the same and changing the loading or the general size the same. And then, as you can imagine, if you can go to different systems if you require a lot more drug for certain applications. And then here on the left is just a GIF showing how dyes release sort of disperse. This is in vitro data, obviously, but you can see how quickly the dye sort of bursts out of the particle. On the right here, you can see this is sort of our what we call our in vivo release library. And what we do here is we have different materials or different materials from the PLGA library, and we have, this is cumulatively, so we have different release points. As you can see, we have formulations, or rather polymers that will release very quickly in the order of days and weeks. And then as you go across to the right, you can see we start to get some of the polymers that last a lot longer. And so we can have a 6-month time point from a particular polymer combination that we can load with a drug of interest or vaccine. And this is some original data that we've done with OVA, and that's been published. There's a lot more in process that we're going to publish soon, but for now, I'll just show this data. The important here is to show that in red is our core shell particles, and this is the titers, so the immune response of the animals are specific to this particular antigen. And you can see that the red is matching 2 bolus doses, so given at both essentially 0 and then about 5 weeks later, and you can see that the response is essentially equivalent. And in fact, the blue line here, which is closest to our response, is for double the dose. So there seems to be an adjuvancy effect as well related to our particles. And the green dose is the actual equivalent dose given in 2 injections. And again, this is illustrated, down here, in peak titers. So again, you can see that our core shell particles are equivalent in titers to a double dose given twice, and definitely superior to a 2 bolus dose. We also have very nice stability. Most of the stability is in 4 degrees Celsius, but a very nice stability in the particle. The vaccine in this case is dried and embedded in the polymer. And again, I mentioned we were able to publish this in Science, and also, as you all know, the space and controlled release is quite, the patents there are quite well covered, but this was such a novel way of creating pulsatile release that we were able to patent the work as well. We have, and as I mentioned, we're working now, and soon we'll both with hepatitis B and polio vaccine and influenza vaccine, and also now mRNA in these particles, and that work will be published very soon. But we continue to have really exciting data where we show essentially the same non-inferior responses with regard to specific titers to the pathogen when we give 1 injection rather than multiple injections. However, we also wanted to see if this was applicable to other things besides the area of vaccines, and we did some work with the STING agonist, which is an immunotherapy drug. It's a cyclic nucleotide. So again, we used this type of system, and in this case what we did is we chose 3 different polymers that release very quickly. So all 3 release, the pulsatile release kinetics occurred within the first 2 weeks, and in this case we compared different tumor models, or we compared the effect of our single injection to multiple injections in different tumor models. And again, this is just to show that we can fabricate the particles. These are have slightly larger cores, but and we can make them with high fidelity in these large arrays. And so here is basically the experiment. Is we used we have the essentially the one injection. We have 3 injections of in red of the sting agonist, and then we have 1 injection of our particles that are loaded with the drug, and we also have empty particles as well. But the key is to compare the blue to the red, and here you can see that both regard to tumor volume and percent survival, essentially our single injection system matched the multiple injection groups, showing that there's a non-inferiority between the 2 groups, and that you can simply deliver the same dosage with a single injection rather than 3. And what was interesting, obviously, there's advantages to doing this, which is improved patient adherence, and you're able to inject this as well directly into some tumors that might be hard to reach, and that's useful because you don't need to have multiple access to that particular area. What was also interesting is that we saw there was a decreased risk of metastasis in these some of these models. So when we left the particles behind after a tumor resection, we noticed that those animals did not develop metastatic cancer at the same rate as the ones who were given the multiple injections. We believe because the material is retained locally for longer periods of time. And again, this work was published in Science Translational Medicine. So now I'll shift gears a little bit away from the self-boosting system to some of our work with microneedle-based delivery systems. And again, we've used them for many different applications in the area of vaccines, as well as actually on patient medical records. But today I'll focus on our work with the microneedles on delivering mRNA-based vaccines, and in particular, doing some engineering to try to develop decentralized manufacturing. And so, as you all know, that vaccine distribution has been a critical roadblock in this particular pandemic globally. Obviously there are conventional methods that mRNA vaccines have been used to deliver the conventional mRNA vaccine, and that includes sort of formulation and packaging, and then it's shipped and distributed, obviously, and then it's prepared and administered at a particular clinic or location by a healthcare provider. And so obviously this creates some unusual roadblocks. As we know, there was an issue with storage and temperature and getting the shots to the right places and sometimes hard to reach places. In order to really try to mitigate this, we wanted to think of ways that we could decentralize the manufacturing of these vaccines, and also maybe have them in a format that allows for less need of infrastructure, healthcare infrastructure, and healthcare administration. And so one way is this idea of the microneedle patch. And I don't know if everyone's familiar with it, but it's essentially something the size of a Band-Aid, rather the little white part inside, the little gauze inside your Band-Aid, or smaller. And it can have many, many tiny needles on the order of, let's say, 100, so a 10 by 10 array. And that can be applied individually, so you could apply it yourself, or you could have an applicator pen and essentially apply it that way for a few minutes. The drug that's on the microneedles is then delivered. And so the idea is there a way to sort of develop a printer or a printing-based system that could make these patches for you so that you could potentially vaccinate a small village in remote areas. And so that's the idea here is there's a standalone device that we can build where the ink itself would be obviously the polymer, in this case, the water-soluble, essentially sugar-based polymer that could also could make the microneedles, and could also in its ink, if you will, could contain the LNP, the lipid nanoparticle mRNA. And sort of that's the idea. Here on the right are some SEM images of microneedles, and you can see for scale, this is less than about half a millimeter in diameter. Sorry, half a centimeter in diameter, and you can the needles are very small, and they don't go very deep through the skin, so the pain perception is not that strong. The tricky part is manufacturing these, again, because currently they require centrifugation. And so we really focused on how we would design this standalone printer, and can it really make stable mRNA vaccine patches. And so the ink, as I mentioned, is modular. It could be switched out to different things. I'll talk about today a little bit about the mRNA LNPs that we put into these into the ink for this particular printer. But the idea is that you have a cartridge with the ink in it. And of course, that would have to be shipped to different locations, but we are now working with, there are several companies that are working in trying to develop these sort of modular units that can synthesize mRNA vaccines, and then that can formulate them into LNPs. So the idea is that this would go in line with these kind of systems, and could be delivered to smaller villages, or even larger villages or towns or cities, but that are that then could be close to a location for distribution. So the idea is that you have this ink, and it's goes into a tray that has the molds for the microneedles. And the key aspect to it is that we have to develop a way to do centrifuge-free mold filling, so that the polymer that contains the vaccine and can really be pulled into these needles. And if you look here on this video in the center, you can see that we were able to create a system that allows you to pull the polymer solution in through the needles quite nicely without the need for centrifugation, and also then undergo drying, so that you can have these microneedle patches. And here's an example of what that would look like as they come off a tray. If you imagine a paper tray, a large paper tray filled with microneedles. And so part of the printer design has been really understanding the throughput of automated microneedle fabrication. And so the way the printer works is that we have one nozzle right now at this sort of first generation prototype, and then that fills these paper or mold trays rather with the polymer ink, which contains both the mRNA and the ink, and I'll go into that a little more in a second. But the idea is to really have better throughput of the filling and drying, and so the number of patches that can be made per day. The key is really to have more areas in the printer for, once they're filled, to be able to dry. So sort of having different shelves or trays in the printer. And as you can see here, you can depending on the size of the tray and how many microneedles fit on a tray, you could realistically have something like thousands of patches printed during a day, which means that in about a week you could have quite a few patches to vaccinate, a small village or town or a particular location in a city that might need to be vaccinated, doesn't have access to a clinic or a standard type of clinic that can do a standard vaccination. And then also another aspect of this engineering design is how do we ensure that everything is clean and sterilized for use as it's applied, so that when the patient applies it, there's no risk, obviously, of infection. And so there are different ways that we feel we can sort of pre-treat the molds, which are made also out of polymer, the PDMS, so that we can have a degassing technique to, one, expedite the printing process, but then two, also to help with sterilizing and essentially cleaning the molds to ensure that there's nothing in the mold that could get on the tips and be transferred to the person. And then finally, there's different ways to get the microneedles out of the paper tray, if you will, or out of the molding tray. And so there's different ways one can imagine doing that. One is obviously you could just pick it up by hand, or you could pick it up by a very simple arm that can then allow you to move it to a location where then the person applying it, whether that's yourself or somebody who's controlling the printer, can apply it to the person. And so we really, so that was sort of the engineering aspect of this project. And then we really needed to understand better how we can control the stability of the vaccine in this case. And that's tricky and of course dependent on the type of vaccine that's used. The mRNA LNPs are additionally challenging because they're already encapsulated in a lipid nanoparticle, which needs to be preserved for its stability and to preserve its activity and efficacy once it's given to the patient. So what we discovered is that controlling the actual droplet size is critical in these printers to ensure that the loading efficiency of active mRNA vaccines occurs. And so, or DNA vaccines, really any type of vaccine in general, but especially the mRNA ones. And so that a smaller droplet was important, is very important, so that any drying effect is that could sort of push the vaccine to the rim of the droplet as it's dried is eliminated, or is rather within the microneedle array so that it can still be delivered. So that was critical. And then we looked at characterizing the microneedle patches. So as you can see here in the middle, we've done many experiments and sort of batch runs to ensure that mRNA-loaded microneedle patches are correctly formed, meaning that the tips are sharp and that structurally they're sound in order to be delivered. And we also calculated the loading efficiency, in this case of DNA vaccines, to ensure that the vaccine itself is really getting in the tips. And as you can see, there's a distribution, but we generally have a 10 microgram loading efficiency for the patches. And then finally, so really to go over this aspect in the fabrication about how best to load. We looked at two processes. One is a one-step loading where the ink is essentially the polymer that we use, and then also the mRNA LNPs all mixed together in one, and that's added to the mold and goes throughout the mold or microneedles, as you can imagine. And that a two-step process, and it might be clear in the next two-step process, is where we use two inks. One ink contains the mRNA and the polymer, the other just the polymer. In this case, we do a first fill where the mRNA and the polymer are concentrated in the tip. And then there's a second step where it's just sort of more for structural, the backing of the needle and so on, that does not contain the mRNA. That eliminates waste and also concentrates the dose into the tip, which is what gets essentially delivered. So if you look here, I think this two-step process is more apparent, but basically the vaccine ink is added to the molds first, and that gets pulled through this vacuum filling method and drying, and then the polymer backing is added as a second step, so that only the tips contain the active dose, or they contain most of the active dose. And that greatly improved our loading efficiency. So here on the left, you can see the effect of going to a two-step process on loading of the vaccine into the tips. And this was done using a protein on the left. And then here in the middle, we show the effect of using mRNA LNPs. And here on the right, you can see the image where we labeled the we used a dye that's more purple or pink, if you will, and you can see that it's concentrated in the tip of the needles using this two-step process. We further had to make sure that the ink composition was tailored for the microparticles for various reasons. One of them is the skin penetration and fast dissolution. So if you can appreciate here on the left, this is where the needles penetrate the skin, and you can see this particular shape. We looked at different shapes of these needles. A pyramidal shape, basically, when applied for 10 minutes, you can see that most of the tips, a little bit of the shaft remains, but most of the tips are dissolved, which is obviously very important for delivering the vaccine. We also had to ensure that the stiffness of the needles and with the drying is sufficient to penetrate the skin so that none of the needles bend, and in that case would not deliver the dose. And again, we see that the pyramid type base shape was critical in developing needles that penetrate well and deliver the drug. And then finally, we looked at the ink composition, polymer composition. We used two materials, or rather three, but we ended up so PVA, polyvinyl alcohol, and PVP and sucrose were the three materials that are used to form the microneedles. And we've learned a couple of things. The primary thing is that the ink need to be composed of at least 50% PVA. Anything below that composition was not sufficient to form the needles that would preserve the mRNA. In addition to that, the ratio of mRNA or LNP mRNA to the polymer ink, so PVA and PVP and sucrose, had to be significant. So a high matrix to mRNA was rather important in order to really preserve the mRNA in the microneedles. And we think that effect is related to sort of embedding these nanoparticles in a stable matrix over time to prevent aggregation or coalescence of the LNPs. And interestingly, here is some data. So on the stability at 6 months of our formulation. So I'm not going to go into the LNP formulation. We used sort of in the actual mRNA, but essentially we used a classic type of formulation with ionizable lipids, cholesterol, and so on. And we've if needed, we can discuss that, but there was nothing unique there that we did. But the data here is, this is data over 6 months. This is with mRNA encoding for luciferase. And you can see the stability of different formulations. So the IM formulation is a liquid formulation, like the vaccines given today. So they're suspended in saline, and you can see that at 4 degrees Celsius they do pretty well, which is in black, and our microneedles also do pretty well at 4 degrees Celsius. But at 25, what we see is that our microneedles, mRNA encoding for luciferase in microneedles, does really well over 6 months at room temperature. However, the liquid-based formulations used in IM do not do as well and degrade over time. So this was very interesting data to show that we could stabilize in the solid form the mRNA formulations. Again, we also looked at data at for 1 month, about 28 days. Again, mRNA encoding for luciferase, so fresh versus stored in the microneedles at 37 degrees Celsius. And again, we see very good stability under these conditions of the mRNA. And finally, we looked at mRNA encoding for RBD, actually in this case. So this is the receptor binding domain. So it's a shorter sequence of the SARS-CoV-2 surface protein. So it's a shorter mRNA sequence, and you can see here in the black is the pre-boost. So this is just the response, the primed response after the first shot, and then post-boost would be after the second shot. And we have a negative control, and then we're comparing intramuscular injection, which is a bolus formulation, from a microneedle administration with the COVID encoding mRNA. And you can see that we are essentially equivalent to the microneedles are equivalent to the IM bolus formulation, showing that the ability to deliver mRNA vaccines from these patches, printed patches, is very feasible, and at least in this case gives us the same neutralizing titers. And again, we also did, this work was done a little while ago. The virus has this ability to really run ahead of the science, but we looked at various variants, and again, we have the same response, that the immune response is equivalent to whether we give IM injections or the classical route, or we use the microneedle solid patches to deliver the mRNA LNPs. And again, this is a 3-month stability study. And again, just highlighting that the immunogenicity of the microneedle patches stored for either 1 or 3 months shows, very strong responses against the COVID virus. And so I hope with those 2 examples I've highlighted how we use some of the tools to develop self-boosting vaccine systems, and also how we are able to build these sort of less centralized manufacturing printers to deliver vaccines in microneedle patches that can be administered without any sharp waste and without really the need for a trained healthcare worker. So now I want to shift a little bit over to a different example. So these are oral based formulations, not injectable in any way, and also switching to something besides the vaccine. So looking at micronutrients. And again, this is to sort of highlight the need. There are more than 2 billion people worldwide that suffer from micronutrient deficiency. And actually, in many cases, the micronutrient deficiency is accompanied by more than one micronutrient, not necessarily just one. And as all of you know, iodized salt fortification has been very successful in reducing iodine deficiency. It's of course has been widely eliminated, and iodized salt is commonly used across the globe. And so the stabilization and mass fortification with other micronutrients, unfortunately, has not been that successful. And there are many different aspects to it, but one in particular that we were interested is the stability of some of these micronutrients that are unable to be fortified in some of the similar ways that iodine and some of the other ones is. And so when we looked at this issue, there are many different, again, challenges. The specific ones we wanted to address was the ability to co-encapsulate multiple micronutrients. Some actually can interact with each other and cause degradation of one or the other, and can also degrade some of the other food, if you will, that people want to, like probiotics that people need to ingest. And then on top of that, there might be things in the food itself that degrade the micronutrients. So by the time they're actually getting through the stomach and into the small intestine where they can actually be absorbed, they're already inactivated. And so we looked at several things. One is the process of cooking and what that does to fortified micronutrients. And then also storage, which is certainly a big issue, especially in countries where the temperatures are very high and there is a lack of air conditioning or temperature controlled storage. And of course, how we can ensure that we have really good intestinal absorption by addressing these particular points. And so what we use is a basic methacrylate copolymer. So that has amine groups, and the side chain is pH sensitive. So we use this material. It has been used before in the pharmaceutical industry, though not so much in food, as a taste masker, and it has a very good moisture protective barrier, especially in film form. So what we used is we tried to encapsulate 11 different micronutrients. Some are water soluble and some are more hydrophobic. And so we used two different methods with the same material to encapsulate it. In the case of oil soluble or more hydrophobic micronutrients, we would directly encapsulate them in the polymer using a common solvent. Whereas in the more aqueous soluble micronutrients, we did a two-step process or double emulsion, where we first generated what we call a micronutrient seed with an excipient, which was then suspended and encapsulated in the synthetic polymer. And so here is an example of 11 different micronutrients that we encapsulated, and I'll just take you to vitamin A, which is I'll talk the most about. But what you see here in these graphs is you can, if we tested the ability, the release from the microparticles of the micronutrient at both in water at both room temperature and 100 degrees Celsius over a 2-hour period. And you can see in the case of this example of vitamin A, nothing is released. But when the microparticles are placed into simulated gastric fluid, there's an instant, very quick release, which means that the micronutrient comes out of the polymer and is able to be absorbed in the GI tract. And here on the right, you can actually see the particles after 2 hours in boiling water, and you can see that their structure is fully preserved. These are in the order of about 150 microns. And then here you can see the recovery after cooking. So encapsulated vitamin A, you're essentially getting most of it out. But free vitamin A is more or less completely degraded after 2 hours under dose conditions. Similar for vitamin D in green, and again, very similar for vitamin C. In the case of B2, you can see that it does pretty well on its own, but again, encapsulation really brings it to another level. And we actually wanted to ensure that the micronutrient itself was being released. So these are animal studies, and this is the GI tract explanted from the mice after being fed the particles, and the particles had a dye that was pink when it was encapsulated, and then when it was released, it would fluoresce green. And so what you see here is, for example, 30 minutes, you can see this is the stomach, and then the GI tract kind of loops through all the way down to the rectum. And so you can see at 30 minutes, you still have some encapsulated particles in the stomach, but you also have a lot of released dye going through the system. And within an hour, you can see that everything's been released from the particles and is moving down the GI tract and should be available for absorption. And so with that, we then went to do human studies, and what we were able to show there with iron, which is which really requires fast release in the stomach because it gets absorbed in the first part of the small intestine, duodenum. What we were able to show there is that we had relative absorption of encapsulated iron of 71 and 89%. 80% counts as equivalence. And so we were able to show that it can absorb just as well as non-encapsulated iron in human subjects. And then similarly, we looked in more detail at vitamin A because it's quite a difficult vitamin to stabilize. It's very unstable in water and moisture, and also in heat, and also it can oxidize readily. And so what you see here, if you look at the data in red and yellow, those are our formulations. These were also coated with starch to allow better flowability, but you can see that over a 5-hour period in boiling water, free vitamin A completely degrades, whereas our encapsulated ones are preserved. And you can also see that the oxidation is greatly reduced in the formulations that were encapsulated. And more importantly, over long periods of time in storage, you can see that over a 6-month period, our encapsulated vitamin A really outperformed both at 40 degrees Celsius and 75% humidity, and at 15 degrees Celsius over this period, whereas the leading product on the market is this light gray, which is considered the most stable form that's encapsulated. And you can see how much we outperformed, or our formulation outperformed them. And again, stability in water is just superb. as well. And then also we looked at it in bouillon, one potential food that we wanted to fortify with this micronutrient. And you can see this is over a year period of time, how our red and green R formulations outperformed the best out there on the market. And then with that, we did another human study, in this case with vitamin A. And again, we were able to show that the absorption is not compromised by the encapsulation, indicating that we do have release from the particles once the food reaches the stomach and is available for absorption. And finally, just sort of to highlight the engineering scale, commercialization scale of this particular project, we started working on very low scale, a milligram base scale in the laboratory. We did our animal experiments there. Then we had to scale this to about gram level to do some of the additional in vivo work in animals, and then to go to our human studies, we had to go external to MIT and scale to kilogram scale, which worked well with a partner. But really, for this to have an effect in the world in the area of food, I always say with the best-selling cancer drug, we're talking about maybe thousands of kilograms of material. But here with food, we're talking about thousands of metric tons. So it's a really, really large scales. And we have 2 companies now that are developing this into commercialized products. And with that, I'd like to thank everyone for their attention. And of course, the incredible team I have at MIT that works on all these projects. It's been wonderful to work with all of them. So with that, I'll take questions.